About mutations, we talked about mutations at the chromosomal level, things like non-disjunction or deletions or insertions of whole sections of chromosomes. Those are very different because you're looking at multiple genes, most likely, because you're looking at whole sections of chromosomes. When you start looking at those, we call those syndromes because they're a characteristic set of symptoms that are connected to that chromosomal change. But if you start looking at much smaller scale, we move into the genetic disorders. So sickle cell anemia is a great example of this. In the wild type beta hemoglobin, there is a thymine in this position that is shown on the picture. But in the mutant, there is an adenine. That in turn changes the mRNA, which changes the amino acid sequence. So we went from a polar structure to a non-polar structure. So we went from something that favored water within its shape and forming and folding to something that didn't. So it folds inwards instead of outwards, creating that sickle cell shape. This is an example. It's called a point mutation. It's just a single nucleotide pair that is changed and then in turn has an effect. This then leads to many body effects, as sickle cell is known to do, but it was just a single amino acid. So it doesn't really take much of a change for these things to occur. Conventions for genetic mutations are very similar to those of DNA and chromosomal errors, but the most basic type is going to be a substitution. So a nucleotide pair substitution is the replacement of one nucleotide and its partner with another pair. Now there's a couple of different results that this can have. Because of the redundancy, because of wobble, some mutations, while leading to a change, will not have an impact on the amino acid, and we call that a silent mutation, as you can see on the middle left. If it changes the amino acid, we call it a missense, and if it changes the amino acid to a stop codon, we call it a nonsense. So overall, that mutation is fairly straightforward. You could also have insertions and deletions at this level. These are called frame shift mutations in general because they lead to a change of the reading frame, unless, of course, it occurs in multiples of 3. Errors during this replication can lead to these substitutions. Mutagens can lead to these substitutions. Teratogens, which lead to birth defects. Carcinogens, which lead to cancer. But these insertions happen. Now, one insertion could be sort of accommodated for, like a single amino acid, maybe leading to silent mutations in some cases, but they can be much more extreme. So like on the picture on the left, there is a frame shift that causes an immediate nonsense. On the picture on the right, there is a frame shift that causes everything downstream to be missense. Who knows when we're going to get to the next stop codon. So really, it's multiples of 1 or 3 can sometimes be accommodated. Multiples of 2 are going to be the worst, but any of them could lead to frame shifts. Now, sometimes mutation is a good thing. Mutation pressure can change the phenotypes. If they occur in germ cells, they can give rise to gametes that can then be passed on from generation to generation. And while the vast majority of such changes are harmful, a very small amount of them can be beneficial. So just because it's mutation doesn't mean it's a bad thing. Duplication and rearrangement and mutation contribute to genomic evolution, with the earliest forms of life likely having a very minimal number of genes, only those necessary for survival and reproduction. The size of the genome has changed over time, has increased over time, with extra genetic material providing raw material for diversification. Accidents during meiosis, errors, mutations, these variations can persist and lead to change. Scientists compare the chromosomal organization of different species to make inferences about the evolutionary processes shaping chromosomes. For example, comparing the human chromosomes to those of the mouse, as we can see on the right, shows that the genes that we have are similar, but that does not necessarily mean that they are on the same chromosome. Blocks of genes on the human 16 are found on 4 different mouse chromosomes, indicating that during the evolution of these processes, the genes in each block stayed together even if they didn't stay in the same place or the same chromosome. Researchers performed comparative analysis between humans and a variety of mammals, and they found many duplications and inversions, and the rate of these events seems to have accelerated about 100 million years ago, as large dinosaurs became extinct and the number of mammalian species increased rapidly. So there's all sorts of changes that can occur, and you can see a variety of examples here. Chromosomal sequences are very important for evolutionary pressure, but they're also important for speciation. Due to chromosomal rearrangements, two populations that have different chromosomes can no longer mate with each other, and that's a step on the way to becoming separate species. Another pattern with medical relevance is that chromosomal breakage points associated with rearrangements are not randomly distributed. Specific sites are used over and over and over again, and these hotspots correspond to locations of chromosomal rearrangements associated with congenital disease. Errors during meiosis can lead to duplication of smaller chromosomal regions. We can have this process happening through crossing over and a variety of other places. So unequal crossing over and template slippage during DNA replication can lead to these changes. We talked about one of these changes before with the gene family for hemoglobin. So it's a series of duplication events that led to the evolution of these genes with related functions. The ancestral globin gave rise to the oxygen-binding muscle proteins myoglobin and to the plant proteins that do similar structures. So there's this superfamily of genes. And after the duplication events, the differences between the genes arose from mutations, and the similarity of the sequences in the genes for both alpha and beta globin proteins support this model. And that can lead to changes on a much larger level as well. So if you compare the genome sequences from different species, there's a lot of evolutionary history here. Exon shuffling being one of those. So we talked about exons being altered, but they can also be moved around, mainly through the help of transposons. But the more similar in sequence the genes and genomes of two species, the more closely related they are. Analyzing highly conserved genes in distantly related species can help clarify evolutionary trends. And genes that evolved a very long time ago can still be surprisingly similar in different species. And biologists have identified a number of genes that are evolving faster in humans than in other organisms like chimpanzees and mice. And these genes are involved in things like the defense against malaria and tuberculosis. And the genes that are evolving the fastest in humans are transcription factors. But there can lead to lots of shifts. And sometimes those genes control other genes. They are master regulatory genes. We can see an example of that here with HOX. So the HOX gene can be expressed very differently in different organisms. You can have them in different amounts, but changes in these HOX genes are one of the major sources of variation within organisms. So we can see on the right, different numbers of the same HOX genes in different organizations and places lead to very different physical expression. So master regulatory genes tend to have ripple effects because they are genes that regulate other genes. HOX or homeobox being one of the more common examples of this. Other differences can come from differences in expression and regulation, particularly of developmental genes. The fossil record tells us what the great changes in history of life have been and when they occurred. Things like plate tectonics and mass extinction, adaptive radiation explain how they occurred, but the genetic mechanisms are we're still trying to figure out. It's a broader field of evolution and devolution called evo-devo. One of these things that have come out is what's called heterochrony. So basically, instead of changing the gene, you change the timing of developmental events. Slight changes in the relative rates of growth can change the adult form substantially, like with the skulls of humans and chimps. They start off fairly similar, but it changes over time due to different parts growing at different rates. So heterochrony alters the relative timing of those reproductive and non-reproductive developmental rates. Other things that can happen are things like paedomorphosis. Paedomorphosis is when juvenile structures are retained. So an axolotl is a great example of this, with the external gills still being expressed. Now, they can have a gene error that leads to the loss of those gills and they start looking like a regular salamander. So that's something that's happening more often now as they've become slightly inbred as part of the pet trade. But combine this with things like homeotic genes, and you're going to get changes like with the bat The homeotic genes determine the basic features as to where a pair of wings and all that kind of structure happens. So if you combine that with changes in timing, we can end up with the same bone pattern that you see in other mammals, but growth and structures being different. So these things all combine together, and changes in sequence or regulation of developmental genes can result in massive morphological change that can be beneficial or harmful. It really depends. So a change in the regulation of developmental genes may be less harmful than a change in a gene sequence, but it's hard to predict. This line of reasoning has led researchers to suggest that changes in the form of organisms often may be caused by mutations that affect developmental genes, the regulation of those genes, and not their actual sequence. Great experiment that has been done looking at the changes of genes and what led to the broader structural differences was done with stickleback fish. So you can see an example of a stickleback here, and they have these ventral spines at the base. And this was being researched by David Kingsley and his colleagues, and he had identified a key gene called PITX1. And this gene influences whether three-spined stickleback fish have a set of large ventral spines. And his hypothesis was that either it was a difference in sequence or it was a difference in regulation and expression. So he studied these stickleback fish, and the spines he found were useful in deterring predatory fish, and they're present in marine sticklebacks, but they are absent in lake sticklebacks. In lakes, large invertebrate predators such as the dragonfly capture juvenile sticklebacks by grasping their spines. This is when the dragonfly is in the larval stage and is also swimming. So they tied a reporter gene, this color growth, that expressed wherever PITX1 was happening. And he found that the genes were being expressed in both the mouth and in the growth of the ventral spines. But for the lake stickleback, the gene was still being expressed in the mouth, but they did not develop their spines. So overall, they concluded that, well, if you check the whole sequence, the amino acids are exactly the same. They are identical, but the expression was not. So he found that the expression had changed in lake-dwelling sticklebacks, and the protein coding sequences of the genes were unchanged. Lake sticklebacks expressed in these regions not related to the productions of the spines, such as the mouth, showed us that the gene is not different, but it is the developmental changes that are, which is sort of that key evidence that he needed to show his hypothesis. So overall, we're looking at mutation and regulation. This will be our tie-in to biotechnology, and we'll go from here.